Welcome to Omni Academy. This is Maria, and today we are starting our ninth lecture on the development of eye. In the last lecture, we studied the development of retina, and in today's lecture, we will be studying the development of optic nerve. So, let's start our today's lecture. Let's start the development of optic nerve with the sources of development. So we need two sources to develop optic nerve, but these sources are number one, optic stalk, and number two, axons of ganglion cells. Axons of ganglion cells are present in the center of the optic nerve, and the optic stalk is giving rise to two structures. The inner layer of the optic stalk is giving rise to the supporting cells called astrocytes and oligodendrocytes, and the outer layer of the optic stalk is forming the outer covering of the optic nerve. So these are the sources for the development of optic nerve. So let's now move to the steps involved in the development. So before starting the process of development of optic nerve, there are few things regarding the connection of optic nerve with the brain you must be aware of. So you can see in this diagram that this area is the optic cup, this area is the optic stalk, and you can assume that here is present the brain. So you can see that the optic cup is connected to the brain with the help of the optic stalk. As I mentioned in the second lecture that the optic cup will become the future eyeball. So you can now call it as the eyeball. And as I mentioned in today's lecture that the optic stalk will become the future optic nerve. So you can call in this diagram it as the optic nerve, and this is the brain. So you can now see that how the eyeball, the optic nerve and the brain are connected to each other. So let's now start the process of development of optic nerve. So the first step in the development is the formation of optic stalk. You can see in this diagram that this is diencephalon, this is optic cup or optic vesicle. With the passage of time, the connection of the optic vesicle with the diencephalon narrows down. You can see that this area is the connection between the diencephalon and the optic cup. With the passage of time, this area narrows down and form the optic stalk. So this area in the later life will become the optic stalk. So this is structure of the optic stalk. Optic stalk has two layers. The inner layer, this one, and this is the outer layer of the optic stalk. And between the two layers, you can see there is a lumen. The choroid fissure is also present on the ventral inferior surface of the optic stalk. You can see in this diagram. So this is choroid fissure, and inside the choroid fissure is present the hyaloid artery. So the first step is the formation of optic stalk. So let's now move to the second step. So the second step of the development is the growth of the axons of the ganglion cells through the inner wall of optic stalk. You can see in this diagram that the axons of the ganglion cells are growing to the inner wall of the optic stalk. As more and more axons of ganglion cells pass through the optic stalk, the lumen of optic stalk keeps on decreasing until it is closed. So you can see that in this diagram that as more and more ganglion cells are passing through the inner wall of the optic stalk, they are obliterating the lumen between the inner and the outer layer of the optic stalk. So this is the second step in the process of development. The third step is the closure of the choroid fissure, and that happens in the 7th week of gestation. And as the choroid fissure, this area is closed. You can see in this diagram that now the choroid fissure is closed. As this fissure is closed, the hyaloid artery is entrapped in the center of the axons of the ganglion cells. So the hyaloid artery is trapped in the center, and in the later life, this hyaloid artery becomes the central artery of retina as the choroid fissure is closed. So the closure of choroid fissure is the third step in the development. So let's now move to the fourth step. Fourth step is the obliteration of the cavity or the lumen between the inner and the outer layer of the optic stalk, and this happens by the 8th week because of excessive growth of axon of the ganglion cells through the inner wall of the optic stalk. As you can see in this diagram, that a large amount of ganglion cell axons are passing through the inner wall of the optic stalk, and this excessive growth is obliterating the cavity or the lumen between the inner and the outer layer of the optic stalk. So by the 8th week, the inner and the outer layer of the optic stalk are attached to each other, and there is no space left between the two layers. And that's the final step in the development of the optic nerve. So this is diagram of the optic nerve. You can see in the center of the optic nerve are present the central artery and the central vein of retina. This is the central artery of retina, and this is the central vein of retina. Surrounding the artery and the vein are the axons of the ganglion cells, and the axons of the ganglion cells are surrounded by the layers of the optic stalk. The inner layer of the optic stalk is giving rise to the supporting cells such as astrocytes and oligodendrocytes, and the outer layer of the optic stalk is forming the covering of the optic nerve. So these are the steps in the development of optic nerve. The myelination of the optic nerve starts near the end of fetal period, and it is completed in the 10th week of human life. Optic nerve is considered as a part of central nervous system rather than being a part of peripheral nervous system. It's so because the optic nerve has three characteristics that make it able to be considered as a part of central nervous system. What these characteristics are? Number one, the optic nerve is derived from diencephalon, and we know that diencephalon is a part of brain, and the brain is part of central nervous system. So this indirectly indicates that the optic nerve is also the part of central nervous system. Second characteristic is that the optic nerve is surrounded by three dural layers just like the central nervous system. We know that the brain and the spinal cord are surrounded by the three dural layers, and what these layers are: number one, arachnoid matter; number two, dura matter; number three, pia matter. These three layers are also surrounding the optic nerve. The third characteristic is the presence of oligodendrocytes and astrocytes in the optic nerve. Oligodendrocyte and astrocytes are the glial cells of the central nervous system. Their presence in the optic nerve indicates that optic nerve is also a part of the central nervous system. So these three are the characteristics that make optic nerve to be able to be considered as a part of central nervous system, while the rest of the cranial nerves are considered as a part of peripheral nervous system. As we know that optic nerve is a part of central nervous system, so any disease that is demyelinating or affecting the central nervous system, just like multiple sclerosis do, the optic nerve is also affected or damaged in such diseases. So let me now just summarize today's lecture. So to develop optic nerve, we need two sources. Number one, optic stalk, and number two, axons of ganglion cells. The optic stalk is giving rise to the two structures. Number one, the supporting cells, and number two, the outer covering of the optic nerve. Now let's move to the steps in the development. So the first step in the development is the formation of the optic stalk. Second step is the growth of the axons of ganglion cells to the inner wall of the optic stalk. Third step is the closure of the choroid fissure. And the fourth step is the obliteration of the lumen between the inner and the outer layer of the optic stalk because of excessive growth of ganglion cell axons to the inner wall of the optic stalk. So these are the steps of the development. So this was today's lecture. In the next lecture, we will be studying the development of rest of the structures of the eye. See you in the next lecture.